All right, well, good afternoon, everyone, and welcome to our monthly water seminar series. This is our next to last one, so we'll have one more the third Thursday in May, when we'll have Daniel Livesey, who's a new scientist with the USDA ARS, will be joining us for that one. But today I'd like to welcome Dr. Kiranmayi Mangalgiri. She's been here at OSU since 2021, so got here during the pandemic. And she serves as an assistant professor in biosystems and ag engineering. And her research focuses on the intersection of determining the occurrence and fate of priority organic contaminants of emerging concern in natural and engineered systems, and the development and design of treatment systems for contaminant mitigation in water and waste streams, particularly to facilitate safe and sustainable water reuse and resource recovery. So today, her presentation is really going to be focused on her research on the contaminants of emerging concern and their relevance to water reuse systems. So Karen, I'll just turn it over to you.
Yes, thank you so much. Thank you for that introduction. And I appreciate everybody here as well as online that you're able to join us and listen to some of the work that I have done in the past, as well what we have going on in the lab right now. So yeah, I'm right now an assistant professor in biosystems engineering, but I've had quite a few pit stops before I got here. I got my bachelor's in India in civil engineering. Then I did my master's in Texas A&M in environmental engineering. Then I did my PhD in biochemical and environmental engineering. And then I did a postdoc in a department that was chemical and environmental engineering. And so the idea of putting all these names up over here to just show that the kind of work that we do is very, very interdisciplinary. And although we are all kind of in these departments that kind of tend to get siloed in what they do, the work that we do in water is very transdisciplinary. So as you all over here and as well online come from different backgrounds. So the work that I'm going to talk about mainly is on emerging contaminants and in water reuse. But our lab in general is interested in understanding how these emerging contaminants, how they occur in the environment, what happens to them as they move through the environment. Do they degrade? Do they get sorbed? And if we can design treatment processes that can mitigate the entry of these contaminants into the environment. So that's the overall focus of my research lab. Today's lab is mainly going to talk about the treatment aspect, especially in the context of water reuse. So to kind of get us started, if you aren't very familiar with this phrase, contaminants of emerging concern, it's an umbrella term that we use to refer to all those novel, unregulated pollutants that are occurring in water and wastewater, primarily because of anthropogenic use or the way we use or dispose some of our waste into the environment. Examples of this are pharmaceuticals or personal care products such as sunscreens or the fragrances that we use, or herbicides and pesticides that we use in our gardens. The big one that's making a big name right now is perfluorinated compounds, or PFAS, also known as forever chemicals. We've been using them for about they've been in use for about 50, 60 years now, and they're very, very stable in the environment. And so because of that stability, they're just accumulating in the environment. And, we've been studying these CECs for almost about 30, 40 years now, but a lot of the work with CECs gathered a lot of momentum about 20 years back. And so EPA has this very nice picture that talks about the different sources of PPCPs, which is pharmaceuticals and personal care products. I really like this picture because it does a relatively comprehensive job that shows us all the different sources of some of these contaminants and what happens to them as they move through the environment. For example, we use pharmaceuticals every day on individual use. When you're not feeling well, you'll have a Tylenol. When you go to the hospital, you have prescription drugs. Some of these contaminants can get into the environment through leaking septic tanks, or biosolid application through wastewater treatment plants, or just through agricultural use in farms or in aquaculture facilities. And so these CECs are actually a concern for us because these are now everywhere, right? In the past 20 years, our technology and ability to determine these contaminants in the environment has really improved. And so we find these contaminants everywhere that we go. So this first graph that I'm showing to you on the left is from a study by USGS almost 25 years ago. This was one of the first nationwide studies that USGS did. So they collected samples from 139 different surface water streams. And they saw that CECs were there in almost every stream, right? This is 25 years back. And there was this understanding that maybe our surface waters are contaminated because they receive wastewater, and wastewater is that collection point for all of our waste. And so there was this understanding that maybe these are very relevant for surface water streams, but not necessarily for groundwater. But a lot of studies with the groundwater has also shown that they really occur ubiquitously across the United States. So this is another study. This was a little bit more recent, maybe 10 years back. This one sampled water from 800 different wells and saw CECs in most of them at different concentrations and at lower concentrations than surface water, but they definitely did occur in groundwater as well. So these CECs are ubiquitous and they tend to catch a lot of public attention. You may have seen news pieces like this that talk about fish feminization of aquatic life in streams that are dominated by wastewater effluent. So you have all these fish in a river. That river is receiving effluent from a wastewater treatment plant. But because the wastewater treatment plant is not able to remove all of these CECs very well, the effluent from the wastewater treatment plant has all these CECs, and so that is getting into the river. And because many of these compounds have endocrine disrupting properties, so they behave like hormones, male fish actually were showing this phenomenon where they were growing eggs. Or you had this other observation in Canada where upstream of a river, the female fish population was about 44 to 50% of the population, but downstream, 85% of the population was female. And because the presence of these endocrine disrupting compounds can affect the feminization of these fish. Now, that sounds like a doomsday scenario, but the good news is that, at least in one of these studies, when the wastewater plant upgraded their system and had better removal for some of these contaminants, they actually saw a reversal of this phenomena in about 3 to 5 years, right? So it's not all that bad. It's just that we need to recognize that this happens and we need to improve the way we do some of our things. Here's another study that kind of caught some attention in the last 5-6 years. This is how aquatic life in some of these streams was addicted to illicit drugs or psychoactive drugs. So the first one was in London. The second study was in Czech Republic, and there's similar studies for United States. But what they saw is that fish in these streams tended to hang around where the wastewater entered the river, and they liked to stay there. They didn't really move away. At times, they also forgot to feed themselves. Their hunting capacity decreased because they were just in that, equivalent of what we say is like they were in that trance, in that zone, right? And this is really unnatural behavior. Imagine populations of fish forgetting to hunt and forgetting to feed themselves. And so when some of these fish were taken away into cleaner waters, they actually even displayed withdrawal type of symptoms of very low mobility. And then, of course, in a few weeks, they recovered. But this thing does happen, right? Coming back home for my last example of how these CECs can impact us. This is a study, this is a big, this is recent development in Washington, the state of Washington. For about 10-15 years now, the state of Washington has been trying to figure out why every time it rains, you have these huge populations of salmon dying off. And obviously, when it rains, there's a lot of stormwater generated. The stormwater carries all the pollutants into a river. So they knew it was related to stormwater, but they couldn't specifically identify what about stormwater is really doing or killing off all these fish. So they tested for 10 years, they tested dissolved oxygen, temperature, heavy metals, suspended particles, dissolved particles, pesticides, herbicides. Nothing really showed or could conclusively get them to why these large populations of salmon were dying off. And about 4 or 5 years back, through non-target analysis, this group in University of Washington at Tacoma, they identified this one compound, 6-PDD quinone, that is causing all the fish kill. Now, 6-PDD, not the quinone, 6-PDD, the parent structure of this compound, is a tire additive. So it is there in almost 80 to 90% of all tires that are manufactured. So this compound, when it reacts with ambient ozone, can form the 6-PDD quinone. So even though 6-PDD by itself doesn't really, have any toxicity, the quinone molecule actually has a very, very high toxicity. And so even though this compound occurs in water at nanogram per liter level, so parts per trillion, very, very low concentrations, it can actually have its LC50 is like in that low nanogram per liter level range. So that's what people figured out is it's related to this structure over here. And so some fish, like this rainbow trout, is very sensitive, whereas some are a little bit more tolerant, but this is what is really tied to all those salmon kills. And so now EPA is kind of trying to see what are the alternates that we can use and how can we start regulating. And so all these examples I wanted to show you was just to establish that these CECs are everywhere. They're very relevant to our water quality, and they're very relevant to the way we use some of our resources. In today's talk, I wanted to talk about how these CECs would move through a circular system and how we can design treatment strategies for making circular systems a little bit more sustainable. So the example of circular system that I wanted to use was water reuse. So if you're familiar with circular systems, here's an example. Think about the way we use water, right? So we get Have I been unmuted all this while?
No, it
Okay.
just happened.
Okay, oh my gosh, okay, good. Thank you for letting me know. Okay, so you have this wastewater treatment plant then disposes of the effluent into a stream or an ocean if you live on the coast. And so this is a very linear way of using water, right? You're taking it from the ground and then putting it into the ocean. That's a very linear way. And the principles of circular economy, under the principles of circular economy, you take that waste stream, which is the wastewater effluent, treat it some more, and recirculate back and reuse it in order to make our systems a little bit more resilient and to reuse our water resources a little bit better. So how that currently looks is the most common way of reusing some of this water is what we call gray water reuse. So you take that wastewater and then you just reuse it on site. That's gray water reuse. But then you have all those CECs, and so gray water reuse is not necessarily the safest way to do reuse. You can also redirect that effluent into an advanced treatment facility and then clean up that wastewater some more and then reuse that water for potable purpose. So if you take that water and charge your aquifer back with it, that's indirect potable reuse. Or if you pipe that into a drinking water treatment plant directly, that's direct potable reuse. And so this is kind of how we do circular water reuse. Currently, the most widely practiced potable reuse is indirect potable reuse. You have fewer instances of direct potable reuse. The problem in the water reuse system is that you have all these emerging contaminants, and so they need to be removed. And so the purpose of this advanced treatment facility is to remove those contaminants that are present in the wastewater so that you can reuse it for potable purposes. And so these advanced treatment facilities are specifically designed to remove these emerging contaminants. The problem is that there are just too many of these contaminants to test out individually. right? Every year, statistics show us that there's like 500 to 2,000 new chemicals that we introduce into our use every year. So if you have 2,000 new chemicals being introduced every year, that's a lot of chemicals to test out individually, right? So that's a big challenge. And you can also see, so these are just some of the structures of the big problem ones that we currently have. If you just look at the chemical structures of this, if chemistry fascinates you, you can see that you have some really small molecules, you have some really large molecules. You have these molecules with a lot of rings in them. You have molecules that don't have any rings in them. And all this variety in the physicochemical structure of these contaminants can make it really challenging. So there's no one universal treatment method that will take care of everything. So our long-term goal is to establish a universal prediction model to predict the fate of these species through some of these water reuse systems. And what I want to focus on in this talk is how light-based processes can degrade these CECs and how we can develop prediction models for these UV-based processes. So light or UV light can degrade these contaminants, whether you're using nature-based systems or whether you're doing engineered systems. So nature-based systems such as lagoons or wetlands are used to polish some of the wastewater before it is reused on site. And part of these nature-based systems is that many of them are open to the sun. So sun has a portion of UV light that can actually degrade these contaminants. And then you have engineered systems that use UV lamps, such as the one shown in the picture below. These UV lamps emit out heavy energy radiation or UV light. And this UV light can again degrade some of these contaminants. So these UV-based systems can also degrade contaminants. So the challenge in trying to get a somewhat universal prediction model is two steps. So there are two independent things we need to do in order to get this universal model. One is to understand how this contaminant behaves in a UV system just by itself. So all of that is in blue. And to do this, we can do some bench scale studies. The second step is to understand how the realistic conditions in an actual wastewater treatment plant or an actual water reuse plant will affect this treatment. And so all of that is done either on a bench scale or a pilot scale. And when you combine data from these two types of experiments, you can come up with a more predictive model that is a little bit more universal. So I'm going to talk a little bit about engineered systems to start with. So just an example of what our bench scale studies for these engineered system looks like. In the picture over here, we have different types of UV reactors in our lab. So this is just one of the UV reactors we have. So what we have over here is you have a UV lamp in the middle and you have this encasing. And then our experimental solution is in this beaker and you have this pump that is circulating the water through the UV reactor system. And so the more you circulate the water or the longer your solution is exposed to UV light, the faster your contaminant will degrade. So we spike the contaminant that we are interested in studying into the solution here. And then over time, we take small samples of water from this big beaker here. And then we analyze that and we see how the concentration changes over time, right? So this is the most basic way to do this type of an experiment. And so this is data that an undergraduate student, Kiran Stewart, he did last summer. This is degradation for a compound called gabapentin. Gabapentin is an anticonvulsant drug that is used to treat carpal tunnel syndrome. So it's one of the most widely prescribed drugs currently because a lot of people are suffering from carpal tunnel syndrome. And so what we see here is that you have this very linear behavior of the compound degradation, which is a very good thing because as an engineer, all I need to do is decide how long should I let this solution be exposed to UV light to get a certain amount of removal, right? So we can do this type of study to calculate how fast that compound degrades. And then we can also do this in several different water conditions. For example, this bar graph here shows how pH of the water can affect this degradation. So we see from our experiments that at neutral pH, you have the highest degradation, whereas at pH 4 and 10, you have slightly lower degradation. And the reason for that is this chemical structure has this OH group and amine group, and so they both can give off those protons, and you have some speciation change in the compound structure. But just using UV light is not nearly enough. So what most water reuse systems do is use hydrogen peroxide in combination of UV. So UV and hydrogen peroxide together is an advanced oxidation process. So basically you have an oxidant like hydrogen peroxide, you are providing additional energy to it through UV light. And in the system, you are then generating hydroxyl radicals. These highly reactive radicals can then break down most CECs. And so you can get a faster removal for your contaminant. So in this graph here, you see when you have no hydrogen peroxide, you have very little removal over an hour. But as you increase the amount of hydrogen peroxide, you're getting an increased removal of that gabapentin compound. So what we can do is we can calculate how fast that gabapentin reacts with those hydroxyl radicals. And this number, this rate constant, is universal. So once you do this type of an experiment for any contaminant, that number is going to be universal. So you don't have to keep running it for your different systems. That is very specific to this. That is very specific to this contaminant, and it's very inherent to its chemical structure. So that is the first set of how we do this. The second set is then understanding how realistic conditions will affect the amount of hydroxyl radicals that are generated. And then when you club the two together, you can generate your predictive model. So what I want to show here is data from some of the pilot scale studies that have been done from Orange at my work with Orange County Water District in California. So just to give you some background on how some of these advanced water purification systems work, they all take wastewater effluent from a wastewater treatment plant, and that is used as the input. And then they use different types of treatment processes, membrane-based treatment, activated carbon treatment, or advanced oxidation treatment in combination with each other to get you very clean water. So the one that I'm going to talk about over here is the very first treatment train that we saw. So this is what is very commonly seen in most water reuse plants in California. So you have a membrane-based system that uses microfiltration and reverse osmosis. And then you have an advanced oxidation system. So the job of the microfiltration and reverse osmosis system is to remove off all of those large, big contaminants. So you have ideally, you would have only water coming out of the reverse osmosis membrane entering your advanced oxidation system. But that is usually not the case. You have these small molecules, such as acetone, caffeine, dioxane. These are very small and neutrally charged, so they can easily bypass a reverse osmosis system. And they can enter the hydrogen peroxide, UV hydrogen peroxide system. So that UV hydrogen peroxide system has to really be designed to efficiently remove all of these type of contaminants. And so I won't get into the details of how we did this study, but we used a bunch of bench scale studies to understand the individual behavior of these contaminants. And then we did a pilot scale experiment to understand how the water quality affects the generation of those reactive radicals that are responsible for breaking the contaminants. And then we put these two together to develop our predictive model. So in this graph here, the background color gradient is our predictive model. This model tells us that the way you use this model is you can run one experiment on your bench scale to know how fast your contaminant reacts with hydroxyl radical. You take that number and you plug it into this system. And then this will give you, so you can predict the behavior of any contaminant, not just these 6 that we tested, but just any contaminant you can predict the behavior of any contaminant in the system. And so those 6 compounds that we tested match very well with our data predictions. So how do we know that? So it's not very clear here, but the color in the circle is our experimental data. And if the color inside the circle matches the background color of our model, that means that we are in a good match. So there are better ways of showing this through graphs, but I thought that this was more visually appealing and more easy to understand. Also a smaller graph. Okay, and so what we want to do, and because this talk, called for demonstrating our research, but also talk about research needs and potential collaborations, one of the things that we would really like to do is to collaborate with utilities and consulting firms that do this type of consulting for water treatment and water reuse, not necessarily just potable reuse, but any type of oxidation-based process that can be, oxidation-based process in a water treatment or a wastewater treatment plant. The other thing that I wanted to talk about is filtration system. Each of these units also use a membrane filtration membrane filtration step in it. For example, in this very first step, you have this reverse osmosis. You have a reverse osmosis here in the second one, sorry. And so you have ultrafiltration in the third treatment step. So each of these membrane filtration steps generates a reject stream. So the way membranes work is you have wastewater coming in. It allows only water and smaller molecules to go through. And so all the rejected molecules are all concentrated in a waste stream. And so this reverse osmosis concentrate is one of the most difficult waste streams to handle and to dispose of, because that waste stream has a high amount of salinity. If you look at the conductivity, it is like 9,000. And the total organic carbon on it is 50 milligrams per liter. That's pretty high. And so, and not just conductivity and the organic carbon, but you have all the toxic chemicals and CECs that are concentrated in it. If the rejection ratio is about 80%, if you think about it, the concentration of contaminants in this waste stream is 80 times more than what is in the effluent, right? So that's a very, very concentrated stream as well. And so it's one of the most difficult challenges that we have. And EPA identifies managing this waste stream as one of the key hurdles in having more established water reuse all over the country. So what we wanted to see is, can we use these UV-based systems for treating these waste streams? Because UV advanced oxidation is so well designed and suited for removing these contaminants in wastewater and advanced water treatment plants. We wanted to see if we can apply them in these concentrate management systems as well. And so we did our bench scale studies with 9 different compounds, tested a bunch of different oxidation processes, tested them in environmental conditions. And we got some results on how some compounds like this atrazine over here is very sensitive, but some compounds like this atenolol is not at all very sensitive to some of these degradation. The key challenge here is that this type of very targeted approach, when you're dealing with like 10,000 different CECs, is not very practical. You cannot test them out one by one. And what we need is, take a step back and use a more like broader approach in trying to see, can we degrade, in trying to track these contaminants through these degradation processes. So we know that wastewater has a lot of organic matter in it. And the organic matter can be used as a proxy for the concentrations of these contaminants. Some of these studies have already been established for wastewater processes, but the organic matter and the waste composition in reverse osmosis concentrate look very different from your normal wastewater. So what I'm showing you here is the fluorescence signature from the concentrate on the left and wastewater influent and effluent on the right. And if you see, the influent has this big fluorescence signature over here. It's slightly reduced in the effluent, but you don't see that in the concentrate at all. So basically what this is telling us is that the composition of organic matter in wastewater is very different from the reverse osmosis concentrate, because it has gone through a lot more processing than just wastewater. So we cannot use the pre-existing studies from wastewater and apply it to reverse osmosis concentrate. So we need to generate specific parameters for these reverse osmosis concentrate waste streams. Okay, so what we did is we used something called parallel factor analysis to decompose these fluorescence signal into 4 independent components. If you're familiar with principal component analysis, it's very similar to what that process is. But these components actually signify some realistic fluorescence of the organic matter. And without getting into a lot of details, in the end, what we were able to show is that the degradation of one of the components, that is the nutrient tracer over here, very well matched with most of the CECs that we tested. Right. So in this figure over here, you see that the correlation is very linear for this nutrient tracer, whereas for UV humic light, which is one of the other components, we don't really have good correlations. The point is that if you are able to track how that nutrient tracer component of organic matter is changing, you can easily predict how contaminants are degrading as well. And doing this online is very straightforward because you're just measuring the fluorescence. So you can put in a fluorescence probe at the set excitation and emission. And then once you track the fluorescence of this particular component of the organic matter, you can use that to back calculate how the contaminants are degrading. And so we've established this for 9 different compounds. And so we can build on this to establish a slightly more universal method. And so in this way, we are able to address some of the key challenges that we have for water reuse, especially in our active or engineered systems. For example, so these are key challenges that the Water Reuse Association identified in their 2022 meeting. And if you see, managing waste streams generated from water reuse facilities is one of those. The other one is building small scale building level water reuse plants. So basically, how do you take those advanced oxidation systems and make them more suitable for smaller facilities and not like a city-wide facility? So some of the work that we've been doing with our engineered system directly contribute to some of these needs that are identified by wastewater reuse. So I think I have about a few more minutes left. And I also wanted to talk about how we can use these same principles of UV degradation and bench scale studies and pilot scale studies to understand nature-based systems, which are also used in water reuse systems. So these nature-based systems are used as a polishing step. And there are different processes in these nature-based systems that can affect contaminant fate. Contaminant fate refers to what is happening to the contaminant. So if a contaminant is volatile, it'll escape into the air as shown by process 1. But most of the time, contaminants get sorbed onto the soil, process 2. Or process 4 and 5 talk about how microbial processes and plant uptake can affect the transport of these contaminants. Excuse me. But the focus of our work here is this process 3, which is talking about how the presence of sunlight and the UV component of that sunlight can affect these degradation processes. So for this study, we had 4 different antibiotics that we tested. We tested antibiotics because of their relevance to public health. The presence of antibiotics in the environment at an elevated concentration can increase antimicrobial resistance. And so we wanted to see how transformation of these antibiotics in the environment can affect the byproducts, and eventually do they also retain that antimicrobial resistance. So this was part of a study that did that. So to just get you up to speed with what those UV processes and how the organic matter can affect this kind of stuff. So you have an antibiotic, it can absorb UV light, it can degrade, right? Like how we've seen before, that is direct photolysis. But if you have some of that organic matter in there, that organic matter can absorb the sunlight and it can create reactive species like hydroxyl radicals, just like your advanced oxidation process. And so you can actually have some enhancement in your degradation just because you have some organic matter there. So that's a very good thing for us. But if you have too much organic matter, that organic matter will screen the light or it will block the light. So that will negatively affect your treatment. And if you have too much organic matter, it can also scavenge some of these reactive species. And so, basically you have this enhancement and suppression of degradation happening at the same time. And so what we are trying to understand is, when does the enhancement dominate the system, and when does the screening and scavenging affect the system? So most of these studies that are performed for environmental fate use Suwannee River natural organic matter as the source of DOM. Like I mentioned before, organic matter properties can change depending on the source. And so to make comparisons between different labs more easy to study, one of the solutions is everybody uses this standardized dissolved organic matter source as a surrogate for organic matter to make results more comparable. And so this is organic matter that is sold by International Humic Substances Society. They collect water from Suwannee River in Georgia, and then they put it through a reverse osmosis system. Then they collect the organic matter that is held on to the reverse osmosis membrane, and then they sell you that, right? So different labs, you can buy 1 gram of that for your study. And so that's what most labs do. And so we did our study in the Suwannee River natural organic matter, which is a standardized organic matter. And so what I'm showing you over here is data for ciprofloxacin. So each of these dots here is giving you the rate constant at different conditions. So when you have no organic matter, you have a very high rate constant. That means you have a lot of removal of ciprofloxacin. But as the organic matter increases, you have this drop, right? So all of these points are our experimental data. And the line that is running through in between is coming from our mathematical model. So our mathematical model is developed from first principles. It's not an empirical model. It is from first principles based on the kinetics of this degradation. And so this is what we see for the standardized Suwannee River natural organic matter. Our question was, you don't see Suwannee River natural organic matter in all the waste sources that you're going to look at, right? So if you have an agricultural effluent dominated stream, you'll see agricultural wastewater there. If you have a wastewater dominated stream, the organic matter will have more wastewater organic matter in it. And so we wanted to see how agricultural waste organic matter can affect this process. Does it look the same in Suwannee River and agricultural waste streams alike? Right, and so this is what we tested. So Suwannee River is what we tested first, and then these 3 poultry litters, we just wanted to do a bench scale study. So we took aqueous extracts of these poultry litter as an example of some of the agricultural waste that we may potentially see. So this is kind of what we see. This is a complicated graph. I'm just going to walk you through it, and hopefully you won't get lost into all of the details. So in these 4 graphs here, the first one is Suwannee River, which is the purple graph, and all the other 3 is degradation in our agricultural waste. Okay, so what you see for ciprofloxacin is that the organic matter from agriculture as well as the Suwannee River kind of have the same effect. But when you look at roxarsone, you kind of see the slightly different behavior. And then the different behavior is even more enhanced for sulfamethoxazole. So if you see sulfamethoxazole here on the very bottom row, you see that in Suwannee River, you have this constant decrease. But whereas in all the agricultural sources, you have this drop, and then you have this increase. So the point over here is that our predictions from some of those standard DOM materials cannot always be used directly for other waste sources. And so if we are trying to look at agricultural waste streams, we need to understand how agricultural organic matter will affect that degradation. And if you're looking at water reuse from a reverse osmosis constant, we need to understand how that organic matter will affect that degradation process. So this is one of the big holes that we currently have, is that we don't understand how organic matter from different sources affect some of these degradation process, not just photodegradation. It can also affect sorption and microbial degradation as well. And so this is one of the big needs that we have. And the reason for this behavioral difference is because especially in these photolytic processes, the steady state concentrations of these reactive species that are generated are very different in Suwannee River compared to the agricultural waste. And that key difference helps us in predicting the behavior of other contaminants in agricultural waste. So if we understand just how different sources are generating their reactive species, then we can plug in the number from our bench scale and just develop. So this is kind of our way of trying to get to that predictive modeling for more universal behavior. And so in today's talk, I mainly focused on the photodegradation of these emerging contaminants, especially in water reuse systems for nature-based as well as engineered systems. But we have like other studies that we do in our lab as well. So we have another graduate student in our lab. She's currently working on environmental occurrence of PFAS, some of those other emerging compounds. And then we have Nathaniel Bolujoko. He's a PhD student. He's working on the fate of antibiotics in phosphorus recovery and other manure handling systems. And so these are some of the work that I do, but they're not necessarily related directly to water, so I didn't talk about them over here. And so with that, I just hope that I was able to show that we do some very bench scale work, but we take this bench scale work and also do, we are interested in doing some pilot and field scale or full scale work. Our effort is in generating some of the fundamental knowledge that governs these contaminants of emerging concern, but we also want to make sure that we take it out of the lab scale, bench scale level and are able to take it to a pilot and full scale level as well. And so that is something that we are very interested in doing. And I have experience in doing those type of projects. And with that, I leave you with this slide again on water reuse, the challenges of water reuse. And I'm happy to take any questions. And I'd like to acknowledge all the funding sources over the years that have supported all this. Thank you. Yes, Doug.
This is a completely smart aleck question.
Please do.
Is there a market if we could develop like an Illinois River standard poultry litter? Could that be a revenue source for you?
I think so. I think so, because every conference that I go to, there is always this huge session on photodegradation and environmental degradation. And this is the big challenge is, that International Humic Substance Society has only like 7 or 8 type of standardized sources that they use and they sell. And they recently ran out of one that they collected from Antarctica. And there's no more of that source. And if they need to go get that, they have to have a whole mission, go to Antarctica, get that, and then purify it and sell it. And the thing is that Pony Lake sample, I think it is Pony Lake, that sample is the closest that comes to agricultural waste. It's not really, because that, I'm told, and I don't know how real this is, but I'm told that the sample that they get from has a lot of penguins around. It is.
More chickens than pigs.
Yeah,
Well, I don't
yeah.
know. I don't know if you get the concentration thing that.
I think this, yeah, they are huge colonies there, and that's the closest source. So the thing is, the history of a lot of this organic matter photolysis is coming from oceanography. Mm-hmm. And maybe some of the more environmental studies. So these wastewater sources and agricultural sources or anthropogenic sources are kind of new, not like 5 years new, maybe 10, 15 years new, but the need for engineering right now is to have those standardized sources for comparison for engineered systems. And so I've been, as a PhD student, I went and asked everybody, do you have any source that is related to agricultural waste? And then I followed up as a postdoc, I followed up as an assistant professor, and we don't.
How much do you pay for a gram of Suwannee River?
It's like $100.
$200?
100, 150, I think. I'm not sure.
5 times price of silk.
Yeah. They sell it to you in like 1 gram small bottle.
1/5. So
I think the key is to standardize it and have enough of it. And so if there are enough takers, because if you go to IHS's website, there's a huge database that they've done the NMR, they've done like FTIR, they've done everything, all the analysis on it. How much metal is there? How much? I mean, a whole bunch of analysis that even I don't really understand. I'm not an organic chemist, but all of that data is there. And people use it for all sorts of experimental analysis, not just photodegradation.
There's a question from online that says, what is the efficiency of the UV oxidation method compared to other methods? And what methods are currently utilized for treating seeds?
So when we think about treatment, there's two types of treatment we think about. One is destructive treatment. So you're taking the contaminant, you're breaking down the structure. And the other type of treatment is you're doing a phase transfer. So it's in water, you're transferring it into maybe, you're using activated carbon or biochar and sorbing it on. And so in the destructive treatment, the UV hydrogen peroxide is the most widely used advanced oxidation process for the destruction. And that's and people have been now trying to look at chlorine-based or UV chlorine as an alternate because chlorine is a lot more cost-effective. And so those are the two big systems in the destructive world that are most efficient. But when it comes to the phase transfer, so you have activated carbon or you have biochar, I think it would be unfair to compare the efficiency of treatment or removal for destructive treatment and phase transfer because they work on two completely different principles, right? So if you have phase transfer, everything is sorbed onto the activated carbon, so you have clean water. But now what do you do with the activated carbon? It still has the contaminant on it. So that has a different cost associated with removal. So, or same thing with membrane, right? So you have a membrane treatment, it is filtering out the contaminants. You have clean water, but you have this waste stream and you don't know what to do with it. So disposal of that waste stream is not counted towards the treatment, but that's part of the, if you do a full LC analysis, that will count towards it. So, yeah.
There's also a question of, are there any regulatory considerations or guidelines specific to the use of UV oxidation for treating CECs? And how do they compare to regulations for other treatment methods?
So in the water reuse world, the regulations are very state specific. So in California, the regulation is that you have to have an oxidation. It doesn't have to be UV oxidation, but you have to have an oxidation step after all of the membrane processes. So towards the end, whether you do ozone or activated carbon, you have to have a UV, you have to have an oxidation step towards the end. And the guidelines at this point, and here I can pull this up, the guidelines at this point are solely based on this one compound called 1,4-dioxane. If I can pull it up. This one compound called 1,4-dioxane. So at this point, the regulations say that if your UV treatment can demonstrate a 0.5 log removal of 1,4-dioxane, then you're good to go. So 0.5 log removal is about 75% 1,4-dioxane. There's a bunch of other requirements as well, but at this point, the 0.5 log removal of 1,4-dioxane is the key standard for UV treatment in the water reuse systems. And then, here, let me look through these questions. How do they compare with other treatment methods? So for water reuse, if you're I think other states have other rules about the newer treatment processes that are used, but I think they all almost always require some type of oxidation towards the end. I think for CCs at this point, this oxidation is really the key step for removal. In Oklahoma, I think that the regulations for water reuse don't have any stipulations for CC removal at this point. They're mainly based on turbidity removal and pathogen removal, but not CC removal, if I'm not wrong. You had a question.
Yes, thank you very much for wonderful presentation. Thank you. So I'm just curious to know whether you guys were able to assess the different DOM, whether it be the Swanee River DOM or the Ag River DOM, which of those that have the highest generation of reactive oxygen species.
Mhm.
And based on those, whether maybe one or two different types were generated, which one were kind of influencing the degradation more?
Yeah, that's a good question. So, here, let me pull up that graph which was this, right? So here, so based on this, this graph over here, we see that Suwannee River actually had the better degradation for a better generation of those reactive species of singlet oxygen and triplet DOM. Those are the ones we looked at compared to the poultry litter stuff. So we think that this is largely because the poultry litter stuff is a lot more aromatic, and it's counterintuitive because aromatic stuff is supposed to increase the generation of these reactive species, but they're also very reactive, right? So the DOM is generating this stuff, but they're also consuming this stuff. And so we think that in here, despite the high aromaticity of the poultry litter, the steady states that are generated for poultry litter are lower because their scavenging is a lot more higher.
Thank you. I have one more.
Yeah, please.
So also, so for the model you guys are going to, you're working on, I think, so my question is, did you just focus on the parent compound of the parent compound of the chemicals, or you also took into consideration the degradation product? Because these two things might be different.
Yeah, no, that's a very good question. So actually, we did look at degradation products in some of these studies and how they also consume some of these radicals. So that is what leads to a more complete picture. So for example, when we looked at our antibiotics in this study, one of the antibiotics that we looked at is ciprofloxacin, and we know that in general in UV systems, fluoroquinolones, which is ciprofloxacin is a type of fluoroquinolone antibiotic. Fluoroquinolones can convert from one to the other. So even I started with one fluoroquinolone, through the UV process, it has formed a completely different fluoroquinolone. So we did do some of those type of studies and also looked at how the degradation of some of these products, what are we forming? Like another compound we looked at in this study was roxarsone. So we know that in roxarsone, so roxarsone is like an arsenic containing compound, and the arsenic moiety actually comes off and you're left with the large organic molecule. So that's roxarsone. So this arsenic moiety comes off and then you're left with this organic molecule. And so you can, so we didn't get to go to the extent of modeling that because the pure if you want to do those bench scale studies, the challenge is you need to get that compound, that degradation product as a pure compound to calculate its rate constant. So you have to either generate it, so you need to have a really good synthetic chemist friend, or you need to pay some company to manufacture that product for you and then run some of the tests. So I haven't done those type of tests, but I know that there are other people who do this kind of work.
Final question.
Yeah, please.
So in the Suwannee River,
Yes.
and the ag.
Mhm.
So which of the degradation process was more prominent? Was it direct or indirect?
That's a good question. I don't remember at the top of my head. I know that I did model it. So here, let me pull up one of these graphs. So for ciprofloxacin, it's all the indirect one. The direct has very little. Oh, sorry, the ciprofloxacin, it's all that the direct has the most dominance. But for roxarsone, that indirect has more dominance. For chlorotetracycline, if you look at the green one, you have this dip and then it goes up. So that's a combination of the two. So yeah, you can, because we have the steady state concentrate time at different times. We know the reaction rate. So that's actually very straightforward to calculate. So I did look at that. It's just it's not something that we delved into for a long time. Yeah.
Thank you.
Yeah.
So
Yes.
just real quick,
Yeah.
it's not really related to the
Yeah.
reuse, but we
Yeah.
were talking earlier today
Mhm.
about Is there development of some easy screening tests for CECs ongoing out there?
Yeah, at this point, a lot of the broad CEC screening tests are biological based or have some bioassay type of application. So ELISA can be used for some quick analysis. So you don't have to collect the sample, put it in an LCMS. ELISA type of kits are used for CECs. In cleaner systems, that fluorescence is used, can be a good surrogate for the bulk CECs, but not individual CECs. Yeah, those are the two that I can really think of, like some of the easier, quicker ways of just calculating overall what is the CEC load that you have. And then you can also do UV analysis. So I think there are some ways where you can kind of use, but that's applicable to relatively clean systems. Because UV spectra is a simpler spectra than fluorescence. Fluorescence has a lot more detail in it.
So what we were talking about
Yeah.
this morning is
Yeah.
we have this well owner water testing program, and
Yeah.
is there any like real easy, simple tools that we could employ to help people know if they have CECs in their drinking water, their groundwater?
Yeah.
So it'd be really clean water.
Yeah. Yeah. I think that there's two challenges in that. One is, are you trying to determine just the general load of CECs, or is there something particular that you're looking at? And so because there are 10,000 different types of CECs, maybe you have to identify priority CECs that you think are important. So if you think PFAS are important, or antibiotics are important, or steroids are important, and then you can use some types of ELISA kit to kind of do that. But ELISA kits are themselves not as, they're not that of they're still expensive. But at this point, the easiest way still is to take the water, bring it back to the lab and put it through an LCMS system. But the thing is, the ease with which we are able to process environmental samples now is a lot better. Because earlier you had to take the sample, put it through an extraction kit, and then put it on your LCMS. Now there's a way you can do it online, so that makes it relatively cheaper and faster. So there are ways to improve that, and that can be done. Yeah, at this point, just the online or like a probe is tricky. I know that there are lab scale studies that are developing microchips that will do this, but I don't know how field ready they are. Yeah, Jeff.
So recently, as you know, the EPA came out with the PFAS regulation. Are there other CECs that are regulated?
Yeah, so EPA has this thing called Unregulated Contaminant Monitoring Rule, UCMR. And every few years, they change the list, and then they kind of update it. So PFAS was on the UCMR 3, it was dropped off in 4, then they picked it back up on 5. And if you look at UCMR 3, 4, and 5, they have some of these antibiotics in them. I think erythromycin is one of the ones that they test. It's still not regulated. A lot of the regulation comes into play when you think of water reuse, because a lot of the regulation is driven by human exposure and human consumption. And so when you're thinking of water reuse or potable water reuse, let me be more specific, that's when a lot of the regulations for contaminants of emerging concern comes in, because when you have reuse of wastewater, you have to make sure these contaminants are out. So that's when you have these regulations come in. But ambient water, you don't have those many regulations. You have them for some types of pesticides. They are already governed because they have an established history of some toxicity. So it's not like none of the pesticides are regulated or none of the herbicides are. There are some that are, but there are some that are kind of upcoming. And so those are, yeah.
All right, well, let's thank Dr. Mangalgiri. All right, well, like I said at the beginning, we'll have one more of these coming up in May, on May 16th, during the noon hour. And we'll have Dr. Daniel Livesey, a new researcher at the USDA ARS, will be joining us. So thank you again and hope you enjoyed it. And we'll see you next time.
Thank you all for coming.